commented on the microbeads that comment and said the bacterial mats, the way they described them, represented things you get. And this is the time of year where they ought to be east of the flower garden, and they're seeing some frequent water. So I'm wondering if it's start wasting kind of thing. But they said
Good afternoon and aloha. Welcome to the fourth and final dissertation defense by the Fantastic Four. The cohort that started in 2010 came in together, developed amazing camaraderie, worked together, created synergies. Just amazing. It's been an amazing experience for me. So today, last but not least for the summer, is Kurt Ingeman. And this will be the last time you'll see me wearing this ridiculous lionfish shirt until Alex Davis defends a year or so from now. Kurt came to us along with the rest of his cohort in 2010. He received his bachelor's degree, magna cum laude, in biology from Humboldt State University in 2006. He was then a sort of a wildlands firefighter for a while, a ecological tech, doing stuff with birds, and also a biological consultant before coming to graduate school. During that time, he realized that he actually preferred to actually do science than to do something related to science. After working in my lab for a few years in the field, doing underwater research, as you'll see, he developed an ear condition that prevented him from further diving. So he shifted midway towards doing theory to complete his dissertation, which I thought was amazingly admirable and shows not only his breadth, but also the depth of his knowledge. Amazing person, but for a better personal introduction, I'm going to give you one of his cohort members, Dr. Ty Kinding.
Okay. Do you already know what's going on? Sorry, Kurt. Okay, so throughout history, there have been many famous cases of alter egos, including the classic tale of Dr. Jekyll and Mr. Hyde, Clark Kent and the heroic Superman, David Bowie and Ziggy Stardust, and today, Beyonce and her alter ego, Sasha Fierce. Now, over the years, you've probably seen all the different faces of Kurt. And here... But I thought most of you were probably unaware of the fact that Kurt has an actual alter ego. So, ladies and gentlemen, on behalf of the Hickson Lab, I'd like to introduce you to Kurt's alter ego, Carlos. So most of us typically think of Kurt as being a mild-mannered professional, and in stressful situations, Kurt always remains calm and composed. But Carlos can't always be trusted to keep it together. Kurt's a talented teacher, and when he speaks, he always explains himself in an eloquent and thoughtful way. And Carlos also has no problem expressing himself. Kurt's passionate about his career, and he's a great scientist. But Carlos hasn't given up on some alternative career paths just yet, including being a professional soccer hooligan, America's Next Top Underwater Model, a pretty princess, a hardworking pool boy, or the next Ricky Martin. Kurt believes in healthy eating, and he's embraced a vegan diet from time to time. But Carlos says, bring on the meat. Kurt keeps his mind and body sharp by staying active and exercising. But Carlos is a fan of the type of exercise that doesn't feel like exercise. Most importantly, Kurt's a family man, and his beautiful family is and will always remain his #1 priority in life. And Carlos, he can't really argue with that. But Carlos does believe that he has more than enough love to go around. We fully enjoyed both Kurt and Carlos through the years, and we know wherever the future takes Kurt, he'll always succeed given his intelligence, thoughtfulness, and kindness. And when he has that perfect balance of Kurt and Carlos, it'll surely be nothing but smooth sailing ahead.
Thanks, Ty. That was perfect. So, getting serious again. Kurt has had a great record since coming to graduate school. He's published 4 papers already. He came in with a National Science Foundation Graduate Research Fellowship, like the rest of his cohort, just an amazing group of people. He was also nominated by our department several times for the Frolander Outstanding Graduate Teaching Award, which is the highest teaching award you can get at OSU. So without further ado, I'd like to have Kurt present to us the effects of introduced predators on prey population and community dynamics. Kurt.
So I've decided to present this as Carlos. No, I'm not going to do that to you. Thank you for the kind introductions. I really appreciate it, and I appreciate everybody being here, and anybody who happens to be watching this on the internet, thank you for watching. So I want to start today right here on this coral reef, because I think as a society, we place a lot of value on protecting natural places like this. And I think that one of the challenges in doing a good job in protecting places like this is understanding how a natural community responds to change, responds to a disturbance. And as an ecologist, we might phrase this as, what is the response of this natural community to a perturbation? And so one of the tools that we use to answer questions like these are food webs. And now this is just sort of a diagram of our community where the species are indicated by these circles, and they're in this network where they're linked by feeding links. These are trophic relationships. And what a food web can do for us is we can track energy flow, where it starts and where it ends up. But importantly, for what I'm going to talk about today, it also allows us to understand a little bit about this community's response to change, and its response and how change propagates through this community, through these interactions among these species. And the type of disturbance that I'm going to focus on today is the introduction of a new species to a community where it didn't exist before. And that's indicated right here. Specifically, I'm going to be talking about a predator introduction. And that means that this species is interacting with its community by feeding on other members of this community. And what we would like to know, in order to better protect environments, is what kind of impacts are we going to experience when we introduce a new predator to this system? How severe are these impacts going to be? So these could range from minor reductions in the abundance of some of its prey, as you can see here, all the way to catastrophic disruption of this system, where we see complete changes in the species composition and altered energy flows, and we have examples of this, of species that have caused major changes to their recipient communities. And so what I would argue is that when we see something like this catastrophic change, what we've seen is a disruption of the stabilizing processes that allowed this community to remain intact prior to the invasion. So that's where we're going to start, and that brings me to the overarching theme of my talk today and also my dissertation, and that's how do introduced predators alter native food webs via their effects on these stabilizing mechanisms. Okay, so you're probably wondering what are these stabilizing mechanisms, and so we're going to start with just stability. And I think that when you listen to the news, when you watch the news, and we hear so much about ecological change that's happening globally, and it is, we have a tendency to think of balance in nature as this fragile sort of system, and importantly, as a very static thing. But I don't think that that is an accurate characterization of stability or equilibrium in natural communities, because what's missing, and yes, I made this in PowerPoint. I'm really proud of this, by the way, and it only gets better from here. So what this sort of analogy is missing are the dynamic processes that allow natural communities to both resist change, but also to return from a perturbation. And so that's what I've tried to illustrate with these springs. And so we're going to use this analogy as a way to continue to think about stabilizing mechanisms, because I think that they emphasize the dynamic feedbacks that are important when we think about stability. So what do we mean? What kind of stability? In ecology, of course, there are a lot of definitions. We love definitions in ecology. And stability has dozens, right? So I'm going to talk about two particular definitions of stability at two different levels of organization. And the first one is at the level of the population. So within a single species, we might call a population stable if it's bounded between extremes of either going to extinction or increasing indefinitely to some arbitrary value. So importantly, a bounded population is a stable population that remains between these two extremes. Okay, so that's our first definition of stability. The second is at a completely different level of organization. We're talking about now the multi-species community. And our definition of stability here is species coexistence over the long term. So these species are interacting, sometimes by eating each other, sometimes by competing for resources. So one definition of stability is the ability of that community to persist without any members of that community becoming excluded. Okay, so now we have these two definitions of stability that we're going to dive into. And I think to understand how an introduced predator can affect these stabilizing mechanisms, we have to focus down in on the species interactions where stability is produced. So we're going to fade out some of our food web and focus right in here at first. So at the population level, it turns out that predation is one way that we can get regulation of prey population densities. And I'm going to tell you how in just a minute. But what we might ask is, if a predator can regulate population densities of its prey within bounds, what happens when we add a new predator to this system? In other words, what's the cumulative effect of a multi-predator system on the prey population regulation? So we might say, does the prey population remain regulated after we've added this novel predator? And on the other side, if we're talking about this multi-species community, when we have two predators that share prey, one of the ways that we can prevent exclusion of either one of those predators is through what's called adaptive defense. And that simply means that this prey is able to allocate defense towards these two predators in a predator-specific way. And I'll explain why later. This sets up a positive indirect interaction between these two predators that prevents exclusion of either one. So it allows this system to coexist through time. So again, we might ask, if one of those predators is novel, a new predator in the system, how does that change this interaction between the two predators, and can native predators coexist with this invader? So I think now we can refine the broad overarching research question about introduced predators and stabilizing mechanisms. And this will sort of give you the structure of this talk and my dissertation in two parts. So in the first part, we're going to use an empirical system, a coral reef, to explore how an introduced predator, an invasive predator, might alter prey regulation. And we're going to do that with some manipulative field experiments that I did in the Bahamas. And in the second part of my talk, we're going to use modeling techniques to explore the coexistence of native and introduced predators to see if introduced predators disrupt this coexistence stabilizing mechanism. So now we have to leave the theoretical behind and go to a real system. So we're going to replace these nodes with real species because now we're entering a food web. This is a very much simplified food web. I should emphasize that you might find on a coral reef. And the new player in the system is right here. It's the Indo-Pacific lionfish. And you can see that it's right in the middle of that food web. Now I hope that all of you were fortunate enough to see the talks by my cohort because they gave really fantastic talks, and I don't want to repeat a lot of what they've said, so I'm not going to give a tremendous amount of background about the lionfish invasion, but I'm happy to field questions about them. But I will emphasize that they were released by humans. This was a human-caused event off the coast of Florida, and they've now spread rapidly throughout the region, all the way down to South America, and as far north as Cape Cod. These are voracious predators. They will eat just about anything that fits in their mouth, which is considerable, up to about half to two-thirds of their size in many cases. And you can see that they're well defended from predation themselves by these venomous spines. So that means that they're not likely to be subject to a whole lot of predation in the invaded range, or for that matter, in their home range. And really importantly for this talk, I want to focus on how they might be unique in the invaded range. So here's a comparison between the lionfish and what we think is probably the most ecologically similar native predator, the Graysby grouper. And you can see that visually, they present very different cues to prey that need to decide if this is a predation threat or not. So if prey are making this choice visually, they're looking at something that they're really not used to. And some research from the Pacific actually shows that they may not be presenting scent cues that prey can recognize. And we know that olfactory, a sensory modality, is really important for reef fish to be able to determine what is and is not a predation threat. So they don't look, and they apparently don't smell like predators. And furthermore, they don't behave like anything else in the Atlantic. So lionfish employ this unique stalking behavior where they wave their showy pectoral fins and actually herd prey into corners, and then they strike really rapidly. This is going to be a video that's slowed way, way down. It's an extremely rapid strike. They flare their buccal cavity in order to create negative pressure and literally suck prey right into their mouth. And so let's take a look at this. And what I want you to notice is how closely this prey fish allows the lionfish to approach before it takes any sort of evasive action. This is just a few centimeters away. Gone. So what this suggests is that prey species may be, for these reasons, may be naive to the threat of predation by lionfish. That is, they may not recognize lionfish as a predation threat, and therefore they can have these really strong consequences that we've seen throughout the invaded range. So if we want to understand, we know that lionfish have strong direct effects. If we want to understand how they affect prey regulation, we have to understand how prey regulation by predators works. So we're going to start with just natives. So here we have a population of coral reef fish, and we want to know, why does it stay regulated within those bounds? Why doesn't it go to zero? Or up to some arbitrarily large value? And it turns out that predation can produce this if predators cause the highest mortality rates when prey densities are high, and less mortality when prey density is low. And so here we have a pretty high density. The predators aggregate preferentially near this. They cause a lot of mortality, but importantly, they move on and aggregate elsewhere when prey densities have decreased. Okay? That allows density to rebound, because without predators around, these populations can grow. So you can see how predation is this negative feedback that changes in population size that causes populations to grow when they're small and to shrink when they're large. So that's our important stabilizing mechanism. Density-dependent predation is what we call it, because it relies on the density of prey. Now, the way that we visualize this is if we have two categories, low density over here, there it is, and high density of prey over here, and we measure how much mortality occurs at the population level, you can see that it's much higher over here. This is the signature of density dependence. Higher mortality at high prey density. Now this is, I should point out, per capita mortality just means the proportional level of mortality that these populations, so accounting for their population size. Now another way that we can look at this is if we have a continuous range of prey density. So now instead of having two categories, we have a continuous range of prey density, and we see a positive slope of mortality. This is the second signature of density dependence, this positive slope between prey density and per capita mortality. And we can break this down just a little bit. Looking at the y-intercept, what's being described here is the amount of mortality that we expect, independent of any changes in density. It's also the amount of mortality that we expect at low prey density. And the second component of density dependence is this slope. This intensity of mortality, intensity of density dependence. In other words, how fast does per capita mortality increase as we increase prey density? And the steeper that slope, the stronger the density dependence is. But I should emphasize that both of these components are important for prey regulation because, as I said, at low prey density here, that's when these populations are the most vulnerable to extirpation, when they're small. And if we have a high intercept, that puts them closer to zero. So the question becomes, what happens to this stable... Yeah, that was ominous, wasn't it? What happens to stabilizing density dependence when we add a new predator? And another way to phrase that is, what's the cumulative effect of both the native and the introduced predator? One possibility is that lionfish themselves cause density dependence, and so they cause the highest mortality at the highest prey density here. If we see this pattern, what we would expect is to observe an increase in the intensity of density dependence, but no change in that intercept. Alternatively, lionfish could cause flat changes across the entire range of prey density that their effect doesn't change, doesn't vary with prey density. In other words, they're adding density-independent mortality. And then we would expect an increase in the intercept, in the magnitude of mortality, but not in the intensity of density dependence. And finally, and this would be the most destabilizing pattern, if lionfish predation causes really intense predation mortality at the lowest prey densities, they could actually reverse the direction of density dependence. And this would be destabilizing, of course, because now, as prey populations become depleted, they experience more and more mortality. So now it's a positive feedback loop. Okay, so we wanted to test these predictions. We're the Hickson lab. This is what we do. We go out into the field and we conduct field manipulation. So I traveled with my lab mates to Lee Stocking Island in the Bahamas to the Caribbean Marine Research Center. Blessed soul, no longer is operational. We love Lee Stocking Island. And I focused on this little beauty, the fairy basslet. Now this is a common prey of lionfish, so that's important. They're also, we care about these, not just because they're beautiful, but they're really important if any of you have aquaria. These are really important aquarium fish. But importantly for my talk, they are these small planktivorous fish. They live in groups on the undersides of reefs where they dart out for passing plankton. That's what they feed on. And then they dive back into holes, tiny little holes in the reef when predators are around. And I took advantage of the fact that we actually know quite a lot about their population dynamics thanks to Michael Webster, who's a former Hickson Lab member. And as you can see, all around great guy, probably enjoying a Mai Tai with his Hawaiian shirt there. And through Michael Webster's work, what we learned is that these populations are independent of each other. That means there's negligible immigration or emigration, which is fantastic, because that means we can treat each one of these populations as an independent statistical replicate in our experiments. And he also showed that they experience density-dependent mortality thanks to native predators that aggregate towards large population sizes of fairy basslet, and that this density dependence does cause regulation of their population size through time. So how did he do this? Well, he found 16 populations of fairy basslet, which he paired by size and similarity, and to 8 of those, he added brand new recruits in order to increase the density of these populations, and he left 8 paired populations unmanipulated, so no change in density. And so that means going out, capturing little tiny fairy basslet. It's actually a lot of fun. Alex and I did a lot of this in the Bahamas. And then releasing them. And then you return over time to census these populations and to see what kinds of differences we find in the mortality patterns. So what did he find? From his 2003 paper, what he showed is that sure enough, fairy basslet experience higher mortality at higher density. So this is density dependence in action. Okay, great. So we've seen density dependence. Now we want to know what happens when we have our new line, or our new predator in the system. So that's an opportunity for me to go back, and I had this really unique opportunity to conduct a before and after study. So I replicated on the identical reefs, on the same reefs that he had done his experiments 12 years before. I went back and did the same manipulations, now in the context of predation by both native predators and lionfish. And I want to emphasize that not only were we able to get at that categorical variable, but because the unmanipulated densities form sort of a range of densities, when we increase, when we released onto populations that were paired for increased recruitment, that created a continuous range of prey densities. And what that allows us to look at is that intensity of density dependence, to use those regression techniques. So it's not just 2 treatments. So this again is Michael Webster's data. And now comparing the post-invasion experiment. So from before to after, the first pattern that you probably notice is mortality is still density dependent. So lionfish have not completely destroyed density dependence. And yet what we do see is when we compare among experiments, that in both treatments, it appears that we've seen an increase in the magnitude of mortality. But what does that mean for that intensity, that strength of density dependence as a stabilizing mechanism? So when we used fairy basslet density as a continuous predictor along the x-axis, and we looked at per capita loss, that's an index of mortality, what we found is, and I should point out that each of these is an individual fairy basslet population. What we found is, compared to these open circles, in the post-invasion experiment, which are the filled dots, what we found was higher mortality, but no change in the intensity of density dependence. So that puts us in a situation like this, where in the post-invasion experiment, we still see the signature of density dependence. It's still there, albeit at higher magnitude of mortality. Okay? Now this was great. This was a really great start because this before and after picture gives us a really clear pattern of a density-independent increase in mortality between the 2 experiments. But what it can give us is an unambiguous picture of how lionfish are changing this pattern, because of course, this before and after design means that there are a host of other things that could have changed in the intervening years between those. We call those confounding variables that we weren't able to manipulate. And so we really want to know, what are lionfish themselves doing? because one of the possibilities is that native predators in these intervening years have increased in density, or that they've increased their predation rates and that they're responsible for this increase. So what's really required, if you want to understand the effects of lionfish themselves, you have to manipulate that factor. Okay, we call that a cross-factor manipulation because we're going to change both the density of fairy basslet, but also the presence and absence of lionfish. And by controlling that factor, we can unambiguously describe differences to the invader. So that was the second experiment. I went back to the Bahamas, this time to a new location, the Cape Eleuthera Institute near Rock Sound, off of the island of Eleuthera. That's still going on, so that's good. It's still a nice place to go dive. And with my trusty lab mates, my cohort, we created at the reef level predator treatment. So what we did is we took 14 reefs and we paired them by size and similarity. And on those reefs, from half of those reefs, we eliminated lionfish by capturing them. And on the other half, we standardized the density of lionfish. So we have high lionfish, low lionfish, or present absent. Now, I'll point out that we left native predators on both of those reefs, but in testing for differences, there was no difference in the predator community or overall abundance or biomass of predators. So that's important to know. The predators were the same on these two treatments. So we've manipulated the predator level. Now I went back and on each of those reefs, I paired fairy basslet populations and conducted similar manipulations to what I described before. So again, we have populations with enhanced recruitment and controls to pair them to. Okay, a few things to point out. We manipulated both lionfish presence and the density of fairy basslet. So we have two factors that we're measuring. And once again, we created a continuous range of prey densities in order to facilitate that, looking at the intensity of density dependence. And finally, this time we also conducted predator observation. So we noted, at larger populations, do we see more lionfish? So are they aggregating preferentially towards large population sizes the way the native predators do? So that was one of our questions. Now, I had much more complicated analysis than the first one, but I'm going to show you, for the sake of time and clarity, I'm going to show you a similar plot to what we saw in the first one. So again, on the x-axis, we're looking at fairy basslet density. On the y-axis, per capita loss. The filled circles are the ones with lionfish, and the open circles are from reefs where we've removed lionfish. And once again, what you can see is that the magnitude of mortality has gone up, but we see no evidence for any change in the intensity of density dependence. So density dependence still exists, but we see more mortality overall in the presence of lionfish. And now we can directly ascribe that to the invader. And importantly, we also observed something that we didn't see in the first experiment, and that's right here. 2 out of those populations, so about 15% of populations only on lionfish reefs, experienced 100% loss. They were completely extirpated. That means the last fairy basslet on the reef was gone in only 4 weeks. So what makes lionfish different? What allows them to, in some cases, eat the last fish on the reef? So one hypothetical mechanism that could describe this involves prey naivete that I mentioned before. And it would work like this. In the absence of predators, in the absence of lionfish, I should say, native predators, like I mentioned before, aggregate preferentially towards these large populations of prey. But these prey are competing for limited shelter space where they can hide from predators and avoid predation. And because of this competition for shelter space, they experience high mortality at low prey density, at high prey density. But importantly, when their densities are low, they experience low mortality. And that's that density dependence pattern, right? And then predators move on when prey density is low. But imagine now if we are looking at lionfish and they're unrecognized as a predation threat, then whether the density of prey is high or low, if they're not recognizing this predation threat, they may be underutilizing the shelter space. So prey naivete might drive high prey mortality, even when their densities are low. Lionfish, in this case, they eat the last fish. And the reason that they can do this is that they're being subsidized. They're a generalist predator. They can eat almost anything they can fit their mouth around. So while they're eating the last few individuals of our target species, they're subsidized by all the other things that they can eat as a generalist predator. So that's one hypothetical mechanism that would fit with the patterns that we saw. And what it means is that lionfish don't have to aggregate just towards high prey density, right? And that fits with our predator observations. We were just as likely to see lionfish where there were lots of prey and on ledges where there were just a few, indicating that they might be uniquely affected at this low prey density when native predators are not. Okay, so we documented these strong effects of lionfish on prey, in some cases causing extirpation of the prey. So you can clearly see that that's going to have effects on native predators, and that's going to be the next part of our talk. But before we do that, let's see if we can answer this first question. Does this novel predator allow prey populations to remain regulated? So I think it's a little bit of a nuanced answer. On one hand, yes, mortality remained density dependent, so that stabilizing mechanism that we talked about is still present. However, we saw higher magnitudes of mortality, and in some cases, that higher mortality drove extirpation. So in a really important way, these populations are not bounded from zero anymore. And so I would argue that this mechanism has at least been weakened by the presence of our invader. Let's go back for a second. So as I mentioned before, anytime that we're changing the abundance or the presence of native prey, that's definitely going to have effects for predators that share this prey, with whom lionfish share this prey. So we need to ask this question: can native predators coexist with the invader, considering the fact that the invader is eating so many of their shared prey? So to sort of get at this question, what we need to do is to talk about this second stabilizing mechanism. And I mentioned before, that's adaptive anti-predator defense. And I'm going to describe how that works now. So we're going to have a few cartoons here. These castles represent defense that the prey uses to reduce predation rates by each of these predators. And they do so in predator-specific ways. So you'll notice that these castles look different because these are unique to each of the different predators. Importantly, this is not a generalized defense against all predation. It works in a predator-specific way. And the reason that this is adaptive is that they can allocate more or less effort towards each of these predators as each of these predators becomes more or less of a predation threat. So let's illustrate that just a little bit. As this predator increases in abundance, it becomes more of a predation threat, and so the prey adaptively allocates more of its defense towards this prey as a higher predation threat. And importantly, unlike our nation, this predator has a limited defense budget. And as a result, it has to reduce its allocation towards this alternative predator. And what that does is it allows predation rates to increase towards this other predator. So as you can see, just like density dependence did, it provides this negative feedback in response to changes in population size and creates a positive indirect effect between these two predators. The increase in abundance of one produces an increase in abundance of the other that's mediated through the shared prey. So that's adaptive anti-predator defense and how it can create stability in this, because that can allow these two predators to coexist on a single shared prey. So we might ask, what if one of these predators is novel? How is that going to affect allocation of defense, and thus how is it going to affect coexistence? And we might consider two different invasion scenarios. So in the first, let's consider a predator that's similar to the native. So they look about the same. but one that has important differences, traits, that allow it to render defense towards it less effective. So sometimes these are things like fangs, bigger teeth, or it might be increased burst speed, or something that allows it to consume prey even when that prey employ their defense. So importantly, they're recognized as a predation threat, but their defense against them is less effective. We call this, I call it, suboptimal defense. And so that's going to be one of our invasion scenarios. Now on the other side, we could have a totally unique predator. You could picture the lionfish here, one that is perhaps unrecognized as any sort of predation threat. And in this case, limited recognition could cause reduced defensive effort towards this predator. Right? So if prey are not able to recognize the predation threat, they might not even employ their defense. Okay? And this is a different sort of scenario, and it could play out differently. We'll call this prey naivete, and I described it before with the lionfish. And both of these two scenarios could result in important implications for the coexistence of native predators. So as Mark mentioned, I was unable to dive halfway through my dissertation, and so we were scrambling for a project. And so I decided to do a modeling project. And so I wanted to model this interaction because it includes some of the things that I'm most interested about introduced predators. And I wanted to do it in this particular way, and that was to use this particular form of a two-predator system called an IGP model, an inter-guild predation model. And in intra-guild predation, a top predator both consumes an intermediate consumer, but also competes with that intermediate consumer for a shared prey resource. So this is the IGP, and we often use R, N, and P for top predator, intermediate consumer, and R. So if you see R, N, and P, this is what I'm talking about. And I wanted to use this model for several reasons. One, when we look at empirical food webs, real webs, and we look at how different things interact, we see IGP embedded throughout. It's a ubiquitous motif throughout empirical webs. And so it's really important. Secondly, it includes within it, nested within it, important ecological interactions. So if we just focus on these two links, we're talking about a tritrophic chain or a trophic cascade. If we focus, ignoring this link, if we just focus on these two, we're looking at an exploitative competition system. And if we just focus on these two links, we're talking about a parent competition. So within IGP are all these other really important ecological interactions that we want to understand. And finally, IGP, it turns out, is very sensitive to the effects of adaptive defense. So we can't get stability very easily with IGP, but one of the ways that we do get stability is through adaptive defense. So it's important for all of these reasons. What this means is that if we want to understand how a novel predator affects broader food webs, this is a key link. Understanding how they change coexistence in IGP systems is this key link to these broader webs. So that's why I wanted to model this way. And so for this project, I traveled no farther than Cordley Hall, right across the way here. This is theory, and you can do it on your computer. And about halfway through this project that I did with my co-advisor, Mark Novak, I decided to call it Monster, Modeling Naivete and Suboptimal Threat Response, because that's what it started to feel like. Very competitive than any sort of modeling. And so what we did is use equations to model the population dynamics of this IGP system and incorporate adaptive anti-predator response in this. That's all we've done. But what we did to extend our knowledge about how a novel predator could affect this is to incorporate a couple of new parameters. And the first, that I called phi, is how we sought to model suboptimal defense. And what this does is to change the relative efficiency of this link right here. How well does this defense work in reducing predation towards a novel predator? Okay? So that's the suboptimal defense side. We introduced a separate parameter that I called rho to incorporate prey naivete. And what this controls is the perceived predation risk that the prey have for their trophic environment. So imagine prey trying to understand where the predation risks are coming from. What this parameter does is it controls how well they're able to recognize the predation risk represented by the novel invader. Okay? And the analysis that we did is called invasibility analysis, where we looked for conditions that allowed for coexistence and where we saw exclusion, particularly exclusion of our native predator, which is what we're interested in. And I should mention that while we modeled both a novel top predator and a novel intermediate consumer, for the sake of time, I'm only going to talk about the results of the novel top predator, but I'm happy to talk about how those results changed when we modeled the other side. All right. So right now, you're about to think in parameter space. So what that means is that we're going to be looking at some coexistence plots. And the important thing to note is the area of the gray, because gray highlights where all three of these species can coexist. That's why it's labeled R, N, and P. In these plots, any place that the consumer is excluded, that will be labeled RP. You can see right there, those two places where that native consumer is excluded. Now we're looking across parameter space, which means that we're looking across regions of productivity and IGP strength. That's the strength of this link. And I'm not going to go into the details for why we use those parameters. Suffice it to say that those are really important in determining where we get coexistence in IGP. So that helps us understand how coexistence changes when we add either suboptimal response or naivete. Okay. So we're looking at this first plot across productivity and IGP strength. And this is the situation where we have ideal recognition and effective responses. That means that there's no naivete, no suboptimal response. This is like a native system, all right? And you can see that across parameter space, there's a lot of coexistence. Now let's look at the effects of suboptimal response on coexistence. And you can see as we go from left to right, we're decreasing the efficiency towards this top predator. We're giving it bigger and bigger teeth, you might say. Now it has traits that really make this an inefficient defense. And what you can see is that suboptimal response annihilated our three species coexistence. Now in this final plot, parameter space is dominated by just this system, the native prey and the introduced predator. So that's the effects of suboptimal response on three species coexistence. Now let's look at the effects of naivete, prey naivete. They no longer recognize this novel top predator. And so now these three panels are showing us decreased recognition of this predator. So worse and worse ability to pick up this as a predation threat. And here you see, yes, there's an increase in the area where the native consumer is excluded. But compared to the first scenario, and these are similar levels of suboptimal response and naivete, compared to suboptimal response, it's a much more modest change in the region of coexistence. So this was a little bit surprising, and it's the first sort of big prediction that we have that this suggests that a predator that is more similar to native predators, but has some unique trait, will have this dramatic effect on native coexistence, much more so than a totally unique predator. Okay, so I think this is an important thing that we can test using empirical systems. Now we might look at another part, another aspect of the interaction here. And so for this plot, what we're going to be looking at is the population densities of all three species, as well as the levels of defense effort towards each of our predators. And so the population densities are going to be color-coded by their colors here, red, blue, and green, and they're going to be solid lines. And the defense effort towards each of these is color-coded as well. They'll be red and blue, but they'll be the dashed lines. Okay, and we're going to go, moving from left to right, we'll be looking from perfect recognition to complete naivete. Now we're just considering the naivete case. So across a gradient of decreasing recognition. So I want to point out some things. Starting on the left, we start here with perfect recognition. You can see that all three species have positive abundances. So they're all coexisting. But as we move across an increasing gradient of naivete or decreasing recognition of this top predator, we see exclusion of our consumer. So that's the first thing to note, consumer exclusion as we get more naivete. The second thing to note is what happens to the level of effort towards defense towards this novel top predator. So as we get more naivete, we get less effort towards defense. And that makes sense, right? Because a prey that no longer recognizes a predation threat is going to reduce the amount of effort that it puts towards defending against that. But the final thing that I want to point out, and I think this was really surprising, is that as we get increasing naivete, decreasing recognition, the abundance of the resource, the abundance of prey actually goes up. So this is pretty unusual. Naivete means it's getting a worse and worse picture of its trophic environment. It's getting a less accurate picture of where predation threats are coming from, and yet it's increasing in its abundance. And I think this points out some of the interesting things about IGP and the indirect interactions that can happen, because what you'll notice is, along the same gradient, as the prey reduce their effort towards this unrecognized top predator, they increase their effort towards the recognized predator. Okay, so now they're increasing effort towards one of their other predators. That could be good for prey population size. But also they're allowing this omnivore, this novel top predator, to increase in abundance. And by doing so, that causes suppression of the consumer, which allows the resource to increase in abundance. So we're getting an indirect effect through this IGP. Okay, again, I think this is an interesting result that comes out of our modeling effort, and something that I would love to be able to test in an empirical system. If we replaced a novel top predator, or a native top predator with a novel one, would we see this same increase in abundance in prey because of these indirect effects? And that's something I'm working on a proposal for right now. So what are the takeaways from this modeling effort? Well, I think we can see that in many circumstances, novelty can cause exclusion of the native consumer. So the stabilizing mechanism has definitely been affected by novelty, by predator novelty. But it's really different, depending on if we're talking about naivete or a suboptimal response. So there's some context-specific differences. And I didn't talk to you about what happens when we make this predator, the one that's novel, but we get completely different results in some cases. Increasing novelty of this predator can actually allow for the coexistence of a top predator that feeds on it. So in that way, novelty can benefit the native species. And so we get really different effects depending on the identity of the predator. So I think we can start to answer our second question. Can native predators coexist with the invader? Well, we definitely saw a reduced area of parameter space. Recall how that gray really contracted. In that way, no. We saw the native predator excluded in many cases as we got more and more novelty of that top predator. But these effects were context specific, and in fact, there were some cases where novelty benefited native predators. So in that case, again, it's a little bit of a nuanced answer. So going back to our broadest question, when will introduced predators have large effects? We've focused in on these, narrowed down on these specific interactions. When are they going to have big effects throughout the web? And how do they do that? Well, we expect them to have large effects when they're effective at low prey density. That's when, from the first part of my talk, we saw that that's when density-dependent mortality patterns are the most effective, when they're a highly effective predator at low prey density, perhaps because of naivete. Secondly, when they elicit a suboptimal response. So we might expect that a similar predator, but with unique traits, is more likely to cause major dramatic effects throughout the web than one that's totally unique throughout its range. And finally, when it consumes its competitors. So there's a distinct advantage when you're competing for prey by having not only a second source of food, but by eating your competitors, right? So that's another time that we expect these large effects throughout the web. And so going back to our coral reef systems and natural systems, natural communities more generally, We know that these face a variety of threats through human-caused climate change, on reef systems, through overharvest, pollution. There are a variety of threats that these systems face. And so it's vital that we not only understand, but also foster resilience in the face of these threats. And so what I hope that my work does in some small way is to give answers that allow us to better manage these systems so that they continue to look like this for a really long time. So I thank you. I have a few other people to thank before I take your questions. I said I wasn't going to get emotional, but you don't know what it feels like until you actually do it. So thank you very much. I'm going to acknowledge some people, and then I will take your questions. We have the room, so we have plenty of time if you want to ask me some questions. So, first, I want to thank my advisor, Mark Dixon, who was very understanding when I told him I wanted to do a modeling chapter. No, this wasn't the case at all. He was actually super supportive. And Mark has been a mentor in every sense of the word. As a teacher and a scientist and a conservationist and somebody who loves nature. And I can't thank him enough for the inspiration that he's given to me. He was even understanding after I sunk the boat. That happened.
Technically, it didn't sink, though.
It didn't sink. It just turtled up and bounced around in the waves for about a week.
Give yourself a little credit.
Thank you, Karen. Thank you, Karen. It didn't sink the boat. We did lose all the equipment. So that wasn't great. Mark, thank you. You've been a tremendous inspiration.
The best part was a phone call. I get a phone call in the middle of the night from the Bahamas. Mark, this is Kurt. We're okay.
That's the best way to start, especially when you've got undergrads whose lives you're responsible for. So I want to thank the Hickson Lab. I can't think of anybody else that I would rather have been stuck on Gilligan's Island with. They have been a tremendous source of help in every time that I've written a manuscript. Their edits have vastly improved it, and if you thought this talk was hard to follow today, you should have seen it 2 days ago before the Hickson Lab streamlined. They've been wonderful friends, and I respect them all as fantastic scientists, and I hope to continue working with them in the future. So thank you, Hickson Lab. I want to thank Mark Novak, who helped me do things that I did not think that I would be able to do. And he did so with grace and patience. And there were times when, as most of you know, he's a pretty smart guy. But there were times when I wasn't smart enough to stay with him. And I would kind of sit there with my jaw open for a little while before I reeled myself back in. And throughout all of that, he was tremendously patient and helpful to me. When I was the most confused. So
you have no idea how many times I had no clue what you were about to
well, you hide it well, then your jaw didn't fall open. And I want to thank the other members of my committee. As I mentioned before, there was a time when I found out that I couldn't dive, when I considered leaving with a master's degree, and actually went into my proposal meeting thinking that I was going to leave with a master's and not finish my dissertation. And they were tremendously encouraging to stick with it and find another way. So I'm really thankful for that. That's changed my life. And I want to mention the department and especially the staff. I'm going to circle a few folks, the 4 T's. I had to copy and paste Trudy in here because I don't know if she was gone that day, but we just covered up a few graduate students. I think they're from the Mengi lab anyway, so we're not too worried about it. And we stuck Kevin in here too, because not only has Kevin been a tremendous DSO, he's the reason that we can even do any of the work that we do. Diving in the Bahamas, he's a dive safety officer. But he doesn't just do that by filling out forms, hanging out. He came out to the field when nobody else would and endured a fairly hellacious field season with us. And he's also a great friend. He even sacrificed watching the Euros, most of the soccer championships that year, 2012, but Germany didn't win anyway, so it wasn't no big deal. So thank you, Kevin, and to all the staff. I want to thank the Ingemans, some of whom traveled really far. I'll point out this rock star came all the way from Australia to see my talk, so I hope you liked it. So, to my dad, where'd you go, Dad? There you are. My dad he gave me the greatest gift that I can imagine by instilling in me a love of nature. That is something that I've taken with me in everything that I've done, and so I really appreciate that. And I want to dedicate this to my mom, who couldn't be here. She passed away. But she provided me the start that I needed, and she was the most loving mother that you can imagine, and so I really appreciate her influence. And thank you to all of you for coming out. I'm overwhelmed by the fact that you made this trip. To my in-laws, yes, you got a slide. Because you've done such a great job of welcoming me into your family and making me feel like a part of your family. And also being excited about my research and asking about what I'm doing. That meant a lot to me. So thank you so much for being such great parents and family. All right, these are my people. This is my family, my pod, as I call them. And they've been a source of inspiration and up for most adventures. I don't think Mia was too stoked about this adventure, but we've done so much together. Believe it or not, this was Corvallis a few years ago. And this is the reason, they're the reason that I do this stuff. It's their world, and my children, it's your world, and I'm handing that off to you. So I hope that I give you a better place. And last and most, my wife. Where's she? Anybody? I promise not to embarrass her, but I do want to say that we truly did this together, and that you've been my best friend and my biggest supporter, and I love you, and let's keep doing it. Let's keep doing it forever. So thank you so much for all that you've put up with. That pod that you saw, she took care of all those folks while I was off diving in the Bahamas. So she deserves unending credit, and I can never pay her back enough. Okay. With that, I will take your questions.
The committee will meet in Dornfeld after we take a few questions from the audience.
I just thought I'd throw this up to show you I wasn't just playing around on my computer. There are real systems that correspond to what we tried to model here. So I thought I'd throw that up. I didn't have time to talk about it today. Questions? Yeah, Liz.
So maybe you mentioned this and I sort of missed it, but I'm curious sort of the time scale at which a prey can change their adaptive mechanisms, and then how that might matter for the sort of implications relative to their lifespan or the lifespan of their fighters.
Yeah, that's a really good question. I didn't have time to describe it. But when we talk about adaptive defense, we could be talking about a behavioral change that could be almost instantaneous. So we're thinking about ecological time, much faster than the time scale of their life, of their dynamics. Or we could be talking, I think our models best correspond to phenotypic plasticity. So this is a great example of that. Here is a tadpole that can, in response to the presence of this dragonfly, can grow this high tail because it allows them, facilitates escape. But in the presence of salamanders and gape-limited predators, it has a phenotypic response, plasticity, to grow this bulgy head, and that allows it to, or prevents it from being consumed. And I think that's the time scale that our models best represent. You could even think about it as on the evolutionary time scale, so even more slowly, trait changes through time in responses to slow changes. And the way that you can tune that adaptive response is a parameter that I mostly left alone. But I also wanted to point out that's another way to model novelty, is perhaps if you were a novel predator, then the adaptive rate would be slower towards that novel predator. And so that's something I'd like to continue with in the future, is I left that parameter constant, but that's something to play with to see how that changes when you respond rapidly to natives, but much more slowly to non-native predators. Yeah.
Great job, Kurt. I was wondering, so it may be because your grouper lion